Matt. I'm a programmer at the LTRI Institute, research branch of Mount Sinai Hospital. And today I'd like to give a nice quick preview of Cytosel, which is an interactive R-Shiny app that myself and a couple of our groups are developing that I think has some really exciting biomedical and life science research applications. So just a quick overview of the points. There are three things I want to go over in this talk fairly quickly. The first one is going to be a bit of our motivation. So I'd like to give a couple of minutes, sort of a crash course on some interesting and applicable ideas in cancer research to sort of set the stage for why we think this application might be useful to the research community. Then we're actually going to load up the app. I'll show some pre-curated data and show some of the features and some of the statistical inference that can be achieved with the application. And finally, I'll end with some of the app specs, a bit of the under the hood stuff, discussing kind of the development process, as well as where we're hoping to take the application development in the coming months. So just a little bit of overview, some overarching principles of the cancer research space. So one of the most important ideas that's come out of the cancer research field in the past few years is this idea that cancers and tumors, much like the human body, are mosaics of different structures and different cell types. For a long time, the research field sort of considered different tumors and cancers to kind of be uniform and homogeneous in nature. If you get a particular cancer type, you're supposed to get a particular course of treatment. And that was sort of the prevailing theory and working model for many years. But with more advanced research methods, we have now understood that this is not the case. Tumors and cancers are incredibly heterogeneous. There are incredible levels of granularity, of differentiation, of specificity that occur, and really does become this mosaic. So this has led to sort of a shift in the way we think about disease treatment, because we now appreciate this heterogeneity and we consider that this diversity in cancer tumors is really going to be pivotal for us to advance things like personalized medicine and targeted therapeutics. Really the idea that for a particular patient, we need to understand the nuance of that heterogeneity to really tailor his or her cancer treatment to be as specific as possible to that tumor profile. So one of the things that we really need to consider is what are the research methods we can use to evaluate this heterogeneity, and how can we use these methods to increase our understanding of how these cancers evolve. So there are a variety of different ways that we can study cancerous tissue, and the two that I'd like to highlight here because they're quite applicable to our application are visual inspection as well as quantitative measurements. So with visuals, we can do things like create very high dimensional images of our cancers. As you see on the right, we can profile them at the single cell level. We can really see sort of the morphology, the visual architecture, tumors organized spatially, what cells are close together, what things interact, and so forth. On top of this, we can also go into each of these cells and measure them quantitatively. We can look at the genetic information and we can really see what is happening at the genetic level. So for example, we can take this tumor tissue, put it through a sequencing experiment, compare it to a normal healthy tissue and see what differences are occurring and are these differences enough to sort of drive this cancerous state. A really important and fundamental idea here is that these two are not mutually exclusive. It's very useful to have both of them run together because any information gleaned from one might be able to recapitulate or confirm ideas from the other. So it's a really important idea that all of these research methods in the cancer sort of research space complement each other. And that's why we often like to link them together and do many types of experiments, something we often refer to as multimodal experiments, getting information at many different levels to increase our understanding. So at the core of these research methods is a very fundamental principle, and that's when we're looking at cancer tumors, we need to identify what's exactly in there. We need to know what are the types of cells that are in our tumor at a given time. How are they interacting with other cells? Are they changing? Are they evolving? Or are they staying the same? So when we're talking about identifying cells, what we do is we can sort of tag or mark different cells in our experiments and track how they evolve, how they change, and what other cells they interact with. Just as I can sort of be differentiated and marked by different characteristics of myself, including my features or my behavior, we can do that as well with cancer cells. There are different markers or labels that we can look for certain cell types, and that really helps us to identify what is exactly happening in our tumor at a given time and then be able to track any of the changes and get a greater appreciation of how these changes affect the disease. So all of this comes together into our goal for building this app is we want to build an application where researchers are able to find these markers, these cell labels that they might need for their experiments, and they can use experimental data that they may have already generated in order to do this. So here's a little bit of an overview of the CytoCell app with some images, some screenshots on the left. So what we've built is an interactive R-Shiny app, which allows researchers to upload their data and interactively build this list of markers, these tags that they might be looking for different cell types. And this can be applicable to a wide variety of different diseases, different cancers, and so forth. We've built in a variety of interactive visualizations so that researchers, as they're going through this process, can explore all of their data, really confirm or maybe question some of the experimental hypotheses that they may have had going in. We've also built in the ability to save analysis locally and re-upload it. So there can be this recursive nature where a researcher may be conducting this process a few times, kind of optimizing this process for a particular data set. And once it looks appropriate to go to the next step, that can be saved and re-uploaded as well. So we also have statistical scoring for all of these processes, and that provides a measure of confidence for the markers that this application sort of suggests to the user, gives them an indication of how well their data set is performing for actually identifying these labels, these targets. So I'd just like to shift over quickly and actually show what the application looks like with some preloaded data. So here you see the landing page for the application. So this will be the first thing that users see. So you can see that there are a variety of configuration and input options here. The user can either upload his or her own data in specific formats that are explained, or browse public curated data sets that we've already sort of put together that allow them to explore some very well-known and classic disease states. So currently we have two preloadable data sets that users can use and sort of go right out of the gate and look at some of the analysis options. Once they've run an analysis, the side panel here will be populated with a variety of panels that allow them to interactively view different plots, different analyses, and so forth. So one of the top ones we have here is actually getting the marker list that they're going to be looking for. So by default, CytoCell will sort of generate a recommended panel based on what is found inside the data set. And this is a very configurable list. These can be moved in and out of the current working space, depending on the user's interest and level of understanding of what these actual targets are. They can also go to different visualization panels to be able to confirm any of the data that they may be seeing and get greater appreciation for comparison. So for example, they can look at different aspects of the data set in an interactive way and really be able to use this to confirm or, like I said, question any of the preliminary results that they may find. And we've built in a variety of very typical visualizations that researchers in the biology field use that we hope they will find useful in being able to do a lot of important exploratory data analysis. We also, as I mentioned, have a metrics page. So what we do here is once we have that list of potential targets for a particular data set, we're going to be running a multinomial logistic regression with a classic sort of training data set split, training and then testing. And what we're doing here is reporting positive predictive value on a neural network version of the multinomial logistic regression. So for the particular data set, the user can go here, look at the distribution of predicted positive scores, and really get an indication or an idea of how well is the application currently able to identify the different cell states in my data set just using the markers that are recommended. So as I mentioned before, at any time, the user can go back here and customize this, either remove some of these markers if they don't think they're appropriate, or add new ones based on prior understanding. So everything is completely customizable and designed to be run recursively. We also have other built-in things like run logs. So the previous 3 runs for a user will be tracked so that all of the configuration options are available to view at any time, really allowing the user to sort of track the progress through the exploration and analysis of a particular data set. So that's just a little bit of a preview of where we currently are at with CytoCell. And I would just like to end off by talking a little bit about what we're doing with it and where we're headed. So for those who are interested in sort of a bit more of the development and coding side, we currently have this bundled as an R package in a private GitHub repository. One of the big project goals that we've had with this app was unit testing. So we think it's really important to maintain proper testing of our code. So we currently have 94% statement coverage using a classic R testing framework. We also have employed the use of continuous integration and deployment. So our current strategy for integrating new features is to merge pull requests, and we have automatic package testing on those pull requests. And anytime that we make changes to our master production branch, we also have an automatic deployment script to a public server. In terms of the future directions, we're really hoping to continue to get new feature requests and UI adjustment ideas from potential users. We would really like to continue to validate the performance of this app on more public data sets. There is an incredible wealth of public data, particularly in the cancer research field, that I think is directly applicable to users. So we want to make sure that our app is continuing to perform as expected on a variety of different diseases. We're also hoping to expand sort of the public server in the coming months. So one of the big goals is to have persistent user accounts and user sessions that can easily be loaded into the server without the need for any intervention for local saving. And lastly, and I think this has been mentioned by a few people here today, documentation is extremely important to our group. So we're continuing to optimize the documentation based off of our experience with the app as well as other users. Things like best practices, the best way to optimize a given data set, as well as troubleshooting, because biological data can be notoriously noisy and difficult to interpret. So we want to make sure that any troubleshooting that users encounter, there'll be appropriate guidelines for how to deal with that. So I'll leave it there. That's just all I wanted to give a little preview of for our app. But here are the links to two of the groups that I'm currently working with. There's some fantastic researchers and students here that are really helping to make this project kind of take off the ground. So I'm not going to name everybody by name, but these two groups are really fantastic. A lot of amazing work in this field is being done here. And if anyone would like to send me any questions about what they've seen today, my email is here as well. So feel free to reach out if you have any questions or queries about the app that we're developing. Thank you all.